name is Sheri, and I'm currently doing a postdoc at KU Leuven University in Belgium, and my talk will be about pesticide exposure that reinforces the stabilizing effect of thermal evolution on predator-prey systems. So I typically focus on these freshwater ecosystems that are exposed to interacting global change stressors, and they are threatening biodiversity. Typically, I focus on pesticides that can enter these freshwater ecosystems via runoff or also via direct spray drift in agricultural landscapes. And next to that, I also focus on global warming because everyone knows that's happening, and it's predicted that the mean temperature will increase, but also, for example, the magnitude of daily temperature fluctuations. There are a lot of studies already focusing on this, on these interacting global change stressors on single species, but it's also important to study this on biotic interactions. So I used this predator-prey model, and I used Ischnura elegans damselfly larvae preying on Daphnia magna water fleas. And it's useful to use this to enhance predictions about the impact of these interacting stressors to predict actually what will happen with the local persistence of species. So I already told you that these global change stressors can interact with each other, and it can also be synergistically so that they enhance each other's effects. What would be the underlying mechanisms? Both stressors increase energetic costs for the organism. So for example, when they're exposed to a pesticide, there will also be a detoxification cost, and also more energy is needed for repair mechanisms. And also when they're exposed to a higher temperature, there is a higher energetic cost for basal metabolism. Associated with this higher metabolism, there will be a higher pollutant uptake, and there will also be a higher metabolic conversion of original compounds into more toxic metabolites for some pesticides. And this is valid for the organophosphates, and I used the pesticide chlorpyrifos, which works on the nervous system by inhibiting the acetylcholinesterase enzyme. So it causes muscular spasms, paralysis, and eventually death. Thereby, I also estimated the impact of thermal evolution. So these damselfly populations are actually thermally adapted across a latitudinal gradient. And I used high latitude populations coming from southern Sweden. They are then thermally adapted to lower mean temperatures and lower fluctuations in temperatures during the day, while these low latitude populations coming from southern France are adapted to higher mean temperatures and also higher fluctuations in the day. And it's nice because this temperature difference actually matches what will happen in the future at these high latitude populations. So therefore, I could apply a space for time substitution. So with this framework, I could make some assessments. So first, when we would expose these high latitude populations at the cold thermal regime and compare them when we expose them to the warm thermal regime in one generation, we could assess their thermoplastic response to warming. When we then compare the high latitude populations at this warm thermal regime with the low latitude populations at the warm thermal regime, we could assess the thermal evolutionary response. And furthermore, when we would use the high latitude populations at the cold thermal regime and compare them with the low latitude populations at the warm thermal regime, we could assess the thermal plasticity and evolutionary response. So I had these three combinations of latitude and thermal regime needed for the space for time substitution. And thereby, I also exposed them to either a solvent control or a pesticide, 1.5 microgram per liter chlorpyrifos. So I could apply the space for time substitution in the absence and presence of the pesticides. So my research questions were first if warming would magnify the negative pesticide effects on the predator, but also if it would scale up to the long-term stability of this predator-prey system. Furthermore, if also thermal evolution would mitigate the impact of warming on pesticide toxicity, so maybe it could buffer a bit for the higher pesticide toxicity under warming. And if also the stressor interaction type between these two stressors could change across the levels of biological organization, and also if thermal evolution would have an impact on this. Therefore, I set up a big experiment. So I used this damselfly larvae and this Daphnia magna water fleas. I first let them acclimate to the thermal regimes. Thereafter, I exposed them first for 24 hours to either the solvent control or the pesticides at their corresponding thermal regime. And then I exposed the predators for another 24 hours while measuring their metabolic rate. And I also did this for the predation trial. So here, again, they were 24 hours exposed at their corresponding thermal regime. And I used also just one Daphnia magna clone just to make it a bit more easy with this difficult setup already. And I used different Daphnia magna densities to also estimate the functional response. So I got from this, for the predator, the metabolic rate, but also the search rate and the handling time by assessing their functional response. Then I also did a separate experiment because I also needed these parameters to imply in the predator-prey dynamics model. So I also assessed for the prey the intrinsic population growth rate and also their carrying capacity at these treatment combinations. So first, some results for the functional response. So on the left, it's the high-latitude predator at the cold thermal regime, in the middle at the warm thermal regime, and then we have the low-latitude predators on the right. And we saw that the pesticide reduced the predation rate, and this was more strong at the warm thermal regime, so in the middle figure for the high-latitude predators. When we then look at the long-term interaction strength, so we got these results from the predator-prey dynamics model. And this is actually an estimation of the long-term stability of the predator-prey system. So actually the long-term effect of the predator population on the prey population density. So first, we didn't see any effect of the pesticide at the cold thermal regime. So there was no effect. However, when we expose the high-latitude predators also to warming, we see a strong reduction in the long-term interaction strength under warming. So this actually means when we have a lower long-term interaction strength, that the system became more stable. So actually that there was a less chance that both predator and prey would go extinct due to population fluctuations. And this was mainly explained by the higher predator metabolic rate that we also saw under this condition, a decreased predator search rate and increased handling time, while the prey were actually not affected by the pesticide at this treatment. When we then look in the solvent control for first the thermoplastic response to warming, we see for the high-latitude predator that actually the long-term interaction strength increased, so the system actually became less stable. And this was mainly explained by the functional response parameters, so the increase in search rate and the decreased handling time, because the other parameters would actually decrease the long-term interaction strength. When we then also included thermal evolution, we didn't see any effect on the long-term interaction strength anymore. So actually the system became more stable, which is actually quite positive. And this was actually mainly by the increase in metabolic rate that was stronger here than when we looked only at thermal plasticity. Under chlorpyrifos exposure, we didn't see any change in long-term interaction strength when only looking at thermal plasticity. And it was probably mainly because we saw the similar patterns in the prey parameters because they were not affected by the pesticide itself. And they contracted the patterns we saw for the predator. Then when we also included thermal evolution here, we saw a decrease in long-term interaction strength, even stronger than only under warming. So actually the pesticide even made the system more stable. That was a bit surprising. So a little bit of stress is actually good, I think. And this was mainly because the decrease in metabolic rate was here less strong than only under thermal plasticity for the predator. Then I just wanted to show shortly the stressor interaction types that I was talking about between pesticide exposure and warming, and we based this on HSD effect sizes. And I just wanted to show you that we found some differences. On the left, we see at the individual level, so on the level of the predator alone, there were more negative antagonistic and synergistic interaction types found, while at a higher level, when we look at the predator-prey system stability, it was actually positive synergistic. This is, again, probably due because the prey was actually not affected by the pesticide. When we also would include the thermal evolution, we didn't see any change in interaction types, so we found similar ones. However, when we would purely look at thermal evolution, so excluding thermal plasticity, we did see that interaction type could change into more positive ones for the individual level, while it became more negative, at the level of the predator-prey system stability. So it's actually cool that also thermal evolution can change your interaction type, while it's actually more realistic to look at the right column, so including both thermal plasticity and thermal evolution. So for the conclusions, first, it's important to also include thermal evolution next to thermal plasticity to make these predictions, because it enhances the long-term stability of the predator-prey system under the combination of pesticide exposure and warming. Also important to note is that we didn't see any changes in the long-term interaction strength at just 20 degrees with the pesticides, so it's actually, yeah, a remarkable finding, since warming really increased the pesticide effects. And then lastly, that the stressor interaction type changes across the level of biological organization, as we found more negative interaction types, like negative synergistic and antagonistic, at the level of the predator, so the level of, the individual level, and actually a more positive interaction type at a higher level, so at the level of the predator-prey system stability. And moreover, that also thermal evolution could have an impact on this. And with this, I want to thank my funding, the WO, also my previous and current group members, also you, of course, and I'm happy to take any questions. And I still have a little video of a predator preying on Daphnia magna. And he's quite happy, I think, that he got it. Okay, thank you. Yeah.
Great. So just a question. So it seems the predator is now a sit and wait strategy. Do you think if the predator we have more active hunting, the result will be similar?
So it's actually a stationary predator. So it sits and waits until the Daphnia comes by and then.
And but imagine like you want to transfer that to other predators.
Yeah.
Do you think we will have the same type of result? Or?
I think it really depends on the type of predator. And also here, the Daphnia was actually not affected by the pesticide at all. So I think if the Daphnia also would be affected, this would have a big impact on the results.
I actually have a question. So you see negative interactions at the individual level, and then it translates into the positive interactions in the higher order. Why do you think this is happening?
Yeah, I think first because the Daphnia was not affected by the pesticide, but also because it was then less sprayed on. So there was less pressure, I think, of the damselfly. So those interactions would have resulted, I think, in this positive result.
The predator is less, it's not pressing on the
Yes.
prey.
All right, so my name is Justin Van Goor. I am going to start by saying that I'm starting a lab currently. So what I'm going to be showing you today are a system of projects that we are really just starting, right? So observations I've made over the last few years that all kind of tie around evolutionary ecology and community ecology. We study a lot of organisms and we keep accumulating more organisms, but really the glue that holds everything together are worms, nematodes, right? So if you are a student or you have a student or you know of a student that's interested in any of the stuff that I'm going to be talking about today, please feel free to reach out to me. So the reason that I care about worms and the reason that I argue that you and other people should care more about worms is that worms are everywhere, they do everything, and we know nothing about hardly any of them, right? So this tree represents infraorders, right? So these are thousands, if not potentially millions of true species on every tip, right? They're found in every habitat. They have every life history that you can think of. They have really any reproductive history that you can think of as well. You can see some of these tips have names associated with them. Many of them don't, right? So I want to just highlight the point that we really know nothing about this. And this is cool in my world because of job security, I guess. But I think what we're going to find out also is that, like a lot of organisms, nematodes are mediated or facilitated by a system of plastic traits that really help them persist in their environments, right? And I'm going to be spending all day today talking about one genus on this tree, right? So start studying nematodes. So the nematodes I'm studying in particular, I've studied a few, but the ones that kind of hold closest to my heart are fig nematodes, and particularly fig nematodes that are associated with figs from the neotropics, so the Americas. Most figs in the Americas are monoecious, which means that they have male and female flowers within the same individual. They're kind of like sequential hermaphrodites in that way, right? Figs, as you may know, have an obligate pollination mutualism with a pollinator wasp. So this pollinator will enter into a receptive fig. She'll squeeze in, oftentimes popping her abdomen and ripping off her wings. And then she'll pollinate a subset of female flowers that are inside the fig, and she'll oviposit her eggs in another subset of the flowers, right? So true obligate mutualism. About 40% of the time we found, and ranges variably by population and by season, 40% of the time she'll be carrying nematodes, so particularly parasitodiplogaster nematodes. And those nematodes are basically constrained as to the number of individuals that can be in a founder population. We found that if there's any more than 10 individuals in a host, it knocks her out of the sky, right? So there's a lot of selection, there's a lot of pressure to have fewer than 10 in this case. And we see, typically speaking, that this host load is around 4 to 6 individuals, right? So after she's died inside the fig, the nematodes will molt into consumptive adults, and they start to consume the interior of the wasp before spilling out and then molting again into reproductive adults. And they get into these mating aggregates, and then they reproduce, and they lay their eggs around the fig. About 4 to 6 weeks later, little tiny juvenile nematodes are kind of crawling around doing their thing, and then male wasps emerge. So male wasps, in this case, as you can see, look very different than their female counterparts. They don't have eyes. They have jaws that help them break into stuff, but they don't actually have a dietary tract. They're very ephemeral in the case that all they do is seek out females in their developing galls, they inseminate them, and then they release them from the fig, right? If these juvenile nematodes come into contact with a male wasp, they'll consume it, right? And we originally thought that this was kind of a weird thing, but I think that this is not a weird thing because actually it means that it amplifies the local population size for these nematodes, right? So these nematodes, again, are constrained by the low mating group size. This consumption of males and then downstream sort of propagation here allows for them to have many, many individuals that are in the population when female wasps are emerging. So it ensures that their individuals get out into the world afterwards. Right? And then the cycle starts anew. So what I'm going to do today is talk about 5 observations that I've made, that some of which are plastic, some of which you'll find out are not really plastic. And the first of which is kind of this question about the constrained mating group size, right? So you'd expect if most times there are 4 to 6 individuals that are occupying the pollinator wasp, there may be instances in which you have all male mating groups or all female mating groups, right? These are obligately outcrossing nematodes. There's no evidence that we have for self-fertility or anything like that, right? So it seems that, by chance alone, you might expect to see maladaptive sex ratios within these figs that lead to fitness limitation. But we don't see that, right? So when we see individual nematodes inside figs, they're always a female. When we see 2, there's a male and a female. When there's 3, we see typically 2 females and a male, and so on and so forth in this really female bias sex ratio, right? And it seems to, for inbred populations like this, it follows really closely with the sort of theory about how we would expect fitness maximization to happen here, right? Now, our hypothesis for this currently is that this is a density-dependent environmental sex determination mechanism through which the number and the sex of co-occurring nematodes informs the molt, right? So when an individual molts into that reproductive or that consumptive adult, it takes time somehow to figure out who else is around and what sex they are, right, to inform their own sex. And this isn't just a crackpot theory. There's actually some described for a distant nematode in Myrmithidae. This similar thing has been described but not really explored too much, right? So there's some precedence for this. Alternatively, this could be, there was recently described this mouth morphology in an allodiplogaster nematode that can eat other nematodes, right? So possibly the sex ratio is produced by the consumption of excess males within the population. Now, I think that this is probably my preferred hypothesis. I was in Mexico in May. I collected more nematodes, and we were planning on sequencing male and female pools to see if there's evidence of a sex chromosome. If there is, we can restructure this. But now talking about the mouth morphology, if you're familiar at all with nematode literature, you'll know that diplogastrid nematodes, including parasitic diplogaster, are marked by this really weird thing that's called the stomal polyphenism, in which one genotype produces variable mouth morphologies based on the food source that's available to them, right? This is different than what you typically see described, at least in Pristiancus worlds, right? So the Eurysomatus form, if you're associated with a female wasp, you see this really complex mouth morphology in which you have the centralized tooth. This makes sense, right? At least to me as a caveman, you need the tooth to eat the wasp material, right? And then if you're eating those male wasps later on, we tend to see a variably different mouth morphology, to the point that, just to nerd out a bit, Diplogastridae in general is defined by this stomal, or this pharyngeal flap, this idea that there's two stomachs, diplogastrid, right? If you were a conventional nematologist, you would say that this individual is not even in the same family, but it's the same genotype of the same individual, or the same species, right? And the effect is consistent, right? So again, if you're a nematode associated with a female wasp, you have this eurystomatus mouth morphology, and if you're with that male, you tend to see the stenostomatus, a simplified mouth morphology that's more narrow. Now, you may ask yourself, and in fact, I ask myself, how can that work, right? How does something that has no tooth eat wasp material and do so, sufficiently? And we got a pretty satisfying answer when we started doing the genomics of these nematodes, and we found that there's probably a bacterial endosymbiont that belongs in the genus Serratia. It's closest, it's pretty divergent from anything. I think the sequence identity is like 95% with Serratia marcescens, but it's also close to Nematophila, which makes sense. However, I had been seeing this for a long time as a grad student. I didn't know what it was. It turns out it's other nematode people know that they infect nematodes with Serratia and it kills nematodes. In this case, it seems like it benefits the nematode to have it, right? So much like a standard nematid nematode, it seems like they occupy or colonize the gut. It gets defecated out, and it helps break down the chitinous material within the wasp. But also, and the purpose of a grant that I just submitted is, this Serratia probably also kills pathogenic fungus that gets introduced into the fig. There's some precedents to say that that's the case. However, it was unclear to us if this association was obligate or facultative in nature. So where did the Serratia come from? Is it always present? I just last week, my summer student, Jabine McDonald, indicated when I was in Mexico, I collected 70 figs. I plucked out a single nematode from 70 of those figs, right? And she found that every single one of those nematodes had Serratia inside of them, right? So it suggests pretty strongly that this association is obligate, which rules for me because we thus far have not been able to culture these nematodes. But now I think that if they obligately eat Serratia, seeding a plate with Serratia is a good start. So I'm going to do that in the fall. Another observation that we made is that there's hyperfitness following male consumption, right? So if you are at all familiar with nematodes, you'll know that this is a really modest number of embryos, right? So again, they're constrained by that low mating group size. There are few mating opportunities, and as a result, generally defined within my observations, but also previous data suggest, they don't produce a whole lot of embryos at all, right? But if you've gotten into that big mating aggregate and you've eaten a male, you'll see quite a different story here, right? So these things are chock full of embryos, right? Really helps with that sort of local population amplification, right? I studied C. briggsii for a couple of years, and if you mated an individual too much, you saw what we would call a bag of worms, in which basically the gonad wall gets kicked down, the sperm kind of invade everything, they inseminate everything they can. Those embryos molt into juvenile nematodes while inside mom, and this is usually a bad thing, right? So it kills mom, it also kills the individuals because they can't get out. But in the case of Parasitodiplogaster, it seems that they have the ability to break free from mom. So when these hyper fitness events happen, it seems like they have no problem spilling out, and that's how they're ready to go when the juvenile or the female wasps are emerging from their galls. Now, finally, the last observation that we made is that there's a pretty gross difference in the amount of time that this life cycle occurs in, right? So if you have the small mating group that's associated with a female wasp, right, those individuals are walking around, crawling around inside of a fig for 4 to 6 weeks, right, until those male wasps emerge, or sometimes until after the female wasps emerge, right? But if you are involved in one of these mating aggregates, the offspring of that that have to emerge. So they basically have to emerge, they have to infect a female wasp, and they have to ride with that female wasp to the next fig. That whole process only takes 5 to 10 days. A female pollinator wasp only lives for 2 days, right? So it seems like there's a very big difference in terms of the time that these generations may take place, right? And we have no idea how or what's modulating that at all. So in thinking about plasticity in general, I have a couple hypotheses. I'm sure there's a lot more, variation that you could propose as well. It seems that the decisions are all made, like everything is kind of static in the early developmental stages of the nematode, right? And the real trigger is this molt into the reproductive adult, what we call an L4 in the nematode world, right? So in that molt to L4, this ESD decision takes place in which, excess males are eaten, and that sex ratio, the typically female bias sex ratio is produced. At that time also maybe the stomal polyphenism takes place. The level of fecundity, which I don't really think is a plastic trait in and of itself. I think it's just purely how much mating is occurring within a fig. And then the life cycle time, so whether or not you have that long life cycle or that shorter life cycle, right? And the null hypothesis I could have about this is that each one of these things is independently plastic, right? Like there's no effect, the individual makes the decision each and every time, right? Where the serratia association fit into this before, we weren't sure, but now we're pretty sure it's obligate, right? And then the alternative hypothesis that we could have is that, really, it's a cascade, so there's one phenotypic trait, and it's modulated by what you're eating, right? So if you're eating a female wasp, you have this female bias ESD, you have the eurystomatus mouth, you have the serratia, obligate in this case, normal fitness, right, due to the level of mating that you have, and then, your normal life cycle, which is in terms of weeks. And then if you're eating a male, you have this streamline where you have ESD, the simplified mouth morphology, the stenostomatus that really needs that serratia bacteria to eat it. And then you have this hyperfitness due to lots of mating, and then you have a rapid life cycle. So with that, I want to thank a lot of people from all over the world. I want to thank you all for being here, and I would be happy to take any questions that you may have. Yeah, so do you mean in terms of like producing the sexes?
Yeah, exactly. Like the male wasp has more testosterone that is triggering the whole change.
Yeah, right. I'm not sure. It's a really cool. So like another thing, I'm really excited about this idea that we can seed the plates with serratia to try to grow them, right? Most of the work that I do here, I have to go to Mexico to do it, and I work out of a field house, right? So like we're really limited in terms of the sort of experimental abilities that we have, but that's the type of thing that I'd be really excited to do. I think that molt happens. My preferred hypothesis right now is the physical damage that is incurred by the wasp triggers the nematode to wake up, right? So because otherwise it doesn't benefit the individual to start eating the wasp host while she's in flight, right? So I think that this mechanical damage by the wasp has some sort of, or maybe when that mechanical damage happens, hormones from the fig or something happens. But yeah, I think you're right. I think it has something to do with some hormonal thing. Thank you.
Hi, I'm Natasha Gillies. I'm a postdoc at the University of Liverpool in the UK. And today I'm going to discuss some work that we started quite recently looking at the relationship between behaviour and reproduction in 2 species of albatrosses and how that might be shaped by the environment. And I'll just start by acknowledging all our many co-authors and funders on this project. So we know that resources ultimately dictate allocation to reproduction and survival through processes of acquisition and allocation. But we also know that the process isn't quite as simple as that. For one thing, we know that environment plays a role in these processes, but it can be difficult to identify where this acts. So environment can change the availability of resources, the behaviours that individuals use to access them, and it can have direct effects on survival and reproduction as well. And in addition to this, when we're thinking about biparentally caring species, we have to consider acquisition and allocation processes at the level of a pair. And that could be difficult to measure, and it can be even more difficult to analyse. So our aim is to fully resolve these processes in 2 species of albatross across multiple populations to gain some insight into the evolutionary and ecological processes that have shaped them, how they might change in the future. So you might be wondering why albatrosses, and to answer that we have to first think about why seabirds. And actually seabirds can be really useful models for acquisition processes for a few reasons. So firstly, they're very long-lived, so we expect them to exhibit really strong trade-offs in reproduction versus survival. Secondly, they're highly reliant on dynamic ocean systems, which means their behavior and decision-making processes are going to be really strongly influenced by the environment. So the panel I've just brought up here is an albatross example from some work we conducted quite recently, looking at how instantaneous environmental conditions change behavior in wandering albatrosses. And the line to focus on here is the blue one, and basically what this shows is that birds travel more in strong crosswinds. So we know that environment is really driving their behavior. And finally, most seabirds are highly monogamous and exhibit obligate biparental care. And so that means we expect a really strong interdependence of the pair behavior. And so each partner should be quite motivated to minimize costs across the pair as a whole. So our work focuses on 2 species of albatrosses, the wandering albatross on the left and the black-brown albatross on the right. And these are real extremes even for seabirds in terms of their lifespan, their dependence on the environment, and the importance of pair behavior. And perhaps most importantly, we have data sets for these species that cover about 30 years and several hundred individuals. So we have a really great opportunity here to start resolving some of these processes. So I'm going to start by giving a little bit of background on these species and the populations they come from. So wandering albatrosses are biennial breeders with really high breeding success. So they produce 1 egg every 2 years, and they tend to fledge that successfully. Black-browed albatrosses have comparatively lower, more variable breeding success, and they breed once a year, so they lay 1 egg annually. And the populations we focus on hail from the southern oceans. So firstly, we look at Bird Island, which is home to both wandering and black-browed albatrosses. And then we also look at the relatively neighbouring colonies, or islands rather, of Crozet and Kerguelen Island, with wandering albatrosses being based on Crozet and black-browed on Kerguelen. And the reason we compare these populations is because they have quite interesting differences in population trajectory and the environments they experience. So on Bird Island, we've seen population declines for both species over the last few decades. And Bird Island also experiences quite pronounced environmental variation. And in contrast, in Crozet and Kerguelen, we see much more stable population trajectories and relatively less environmental variation. And as well as different life histories, we see quite pronounced differences in foraging behaviour between our 2 species. So looking firstly at Bird Island, there's really, really long looping trajectories from a wandering albatross, and there's more sort of there and back trajectories from a black-browed albatross. And we see similar patterns for Crozet, for our wandering albatrosses, and Kerguelen for our black-browed albatrosses. And the reason I'm flagging this is because these really long trajectories of wandering albatrosses might give them more opportunity to sort of select environmental conditions that might be good for them, and we'll come back to that later. But for now, returning to this flowchart, in our system, it can be quite difficult to measure acquisition processes directly. So what we're trying to do is come up with some behavioural proxies of this. And to start with, we're focusing on this relationship between allocation and reproductive success. And the reason for this is that albatrosses make it relatively easy to measure allocation because they have this really strongly egalitarian biparental care. So what that means is both the male and female make relatively equal contributions to raising the offspring. And during incubation, what this means is they're both taking it in turns to incubate their egg for about 1 to 2 weeks at a time while the other partner is out at sea. And so we can quite precisely measure the investment of both parents. So with that in mind, we came up with some variables that measure time investment. So the first of these is fasting debt. So this measures the amount of time the bird spends sitting at the nest versus the time it spends away from the nest. So the idea here is that If you spend a really long time sat in the nest not eating, then you need to take a proportionally long time at sea to sort of recoup those costs. And we expect that at a pair level, this fasting debt should be minimised for both individuals because neither partner wants to impose a cost on its partner. And then as well as this, we looked at mean trip durations, the amount of time birds are spending away from the nest. And we looked at trip variability, so the variability of the time birds spend away from the nest. So we used 2 different logger types to get at these variables. So we used GPS and then another device called a saltwater immersion logger, which basically tells you if the bird is wet or not. But because it's a bit more straightforward, I'm just going to illustrate our methods using GPS tracks. So these are tracks from a single black-browed albatross. I think there's about 6 trips going on here. And what we can do is look at the time between the bird departing the colony and returning from sea. And that's our time at sea variable. And because we're looking at incubation here and they have this sort of changeover behaviour, if we know when this green bird is back at the nest, we know that its orange partner is out at sea. So we basically get a 2-for-1. We can get the trip durations of both partners. And from these variables, or from this time at sea measurement, we can then calculate our 3 variables. So we have our fasting debt. So here the green bird might be incurring a high fasting debt because it's not spending long at sea for the amount of time it's in the nest. We can look at trip duration. So here the green bird has short trips. And we can look at trip variability. So these would be variable trips. So we then took each of these 3 measures and looked at how they predicted reproductive success, which we measured as a simple success or failure variable, where success is fledging a chick. And we used a Bayesian framework using the BRIMS package in R, using weakly informative priors based on the existing literature. And what that means is I'm going to show you some results that are posterior estimates from those models. And if you're not familiar with that, they essentially quantify the variability and uncertainty in our effect sizes. And if they encapsulate 0, we say there's weak evidence for an effect. So that's basically what you want to look out for. So thinking about our black-browed albatrosses and Bird Island to start with, we have females on the left here, males on the right, and these are our posterior estimates. We see really limited evidence for an effect of fasting debt or trip duration, but it does look like we have a negative effect of trip variability. So what that means is if you go on more variable foraging trips, you are less likely to fledge a chick. But if we turn to our black-browed albatrosses on Kerguelen, we don't see any effects of those behavioural measures. Or rather, we see very weak evidence for effects. So what's going on? So variable trips lead to reduced breeding success, but only on Bird Island. One thing I mentioned at the start is that Bird Island has a much more variable environment. So these panels on the right show differences in some environmental variables between Bird Island in orange and Kerguelen in the sort of teal blue colour. So that's wind speed, direction, rainfall, and sea surface temperature. And you can see that Bird Island is a bit more variable. If it's the case that some birds are simply unable to buffer that variation, that might lead to them having more variable trip durations and as a result reduced breeding success. And if that's occurring at a population level, then it's possible that that might be starting to explain these differences in population trajectory that we've been seeing. Turning now to the wandering albatrosses, so we have the same measures here, and females on the left, males on the right. And again, starting with Bird Island, we have very limited evidence for effect of any of those 3 behavioural measures. If we turn to Crozet, we see a very similar story. So behaviour doesn't seem to have an effect on either wandering albatross population, and that might be a bit surprising because they also experience these contrasting environmental variabilities. So again, this panel on the right is showing that contrast for Bird Island in orange and Crozet in blue. So what might explain that? One possibility is that wandering albatrosses are just very good at buffering environmental variation. So it's possible that because they go on these very, very long looping trajectories, that they're simply able to match their foraging behaviour to their preferred environmental variables or their preferred environmental conditions. So the panel I just brought up here is from some other work we've been doing that looks at the effect of climatic variation on wandering albatross behaviour. And what we essentially found here is that when the Southern Oscillation Index was negative, so this was one of our climatic variables, and that indicates poor foraging conditions for wandering albatrosses. They seem to travel further. So possibly they are seeking out better environmental conditions when ones closer to home aren't so great. So I've talked a lot about speculated a lot on environment here, but one of the key points is that we didn't see effects of pair behaviour, at least using our sort of fasting measure, which is something that we expect to represent those intra-pair decisions. And this is surprising because we expect that partnerships should be highly valuable to albatrosses, and that's for several reasons. So we know that divorce and extra-pair paternity rates are really, really low. We know that coordination is important to them, and we know that partner compatibility is important as well. So that's quite surprising. Some potential explanations. Firstly, maybe there's just low variation in intra-pair coordination and behaviour. If that is really important, then there might not be enough variation for us to actually measure. Perhaps more likely is that pair behaviour becomes more relevant later in the season. So we focused on incubation here, but when the chick actually hatches, then we expect the energetic constraints the parents are acting under to be much stronger. So we will be moving to the chick rearing period later, and maybe we'll see different effects. And finally, it might be that the value of partnerships is experienced elsewhere in their breeding season, perhaps relating to territory defence or some sort of fundamental compatibility measure. So at this point, I've probably raised more questions than I've answered. We've got a lot of work to do, but for now, I will leave you with 3 take-home messages. So firstly, we think that local environmental differences might be driving reproductive outcomes in black-browed albatrosses, and perhaps due to these inabilities to buffer that variation. Second, it's possible that the long-distance travel capabilities of wandering albatrosses make them more resilient to environmental variation if they are better able to match their foraging behaviour to their preferred environment. And finally, contrary to expectations, we couldn't find evidence that pair-level behaviour consequences for breeding success, but it might be that we're not quite measuring the right thing yet. And that is me. Thank you for listening, and thanks to all our funders. Yeah, it's a really good point. It's a big problem with working with wandering albatrosses because, across our data set, you're absolutely right, there's not a huge amount of variation. The strength of this is that we do have just an enormous data set, so we have a bit. But another thing to look at, if we can't capture that sort of failure versus success effect, might be, as you say, to look at chick growth rate or some other measure of investment. We have a bit of data on that, so watch this space.
Hi everyone, my name is Daniela, and today we're going to dive into genomic depths, exploring the genetic diversity of cichlid fishes from Lake Tanganyika through demography and connectivity analysis. The first question that I'm going to ask you is actually, what is demography? So demography is defined as survival, abundance, recruitment, or connectivity between individuals of one population or even more. So we do have many demography parameters that could be considered into a population, such as population size, population density, and population connectivity. But today we're going to focus mainly in population size. But why is even population size important when we are studying organisms and their genomics analysis? So first, to determine its effects on the mutation-driven equilibrium, because of course we know that different populations with different population sizes will be differently impacted by evolutionary forces, and the genetic diversity that will be increased by mutation and lost by fixation on drift will depend exactly on the population sizes. So we know, for example, that drift is negatively correlated with population size, while mutation rate and natural selection are positively correlated. And as a consequence, as a theoretical proposition, we know that genetic diversity is also positively correlated with population size. However, natural populations frequently challenge these theoretical propositions. And in demography, we have plenty studies in the literature describing and studying population size fluctuations. For example, in the Bombovocovarensis B, where they were trying to study the correlation between population size and genetic diversity, and they actually discovered that, well, here you're looking to a population size, a basic population size plot, where in the x-axis you have time and the y-axis population size fluctuations. And here you can see that the population in blue, that is actually more unstable in population size, it's also the population that present a higher genetic diversity. So in this case, they show that instability is generating diversity in bees. But in other studies, that are analyzing, for example, Atlantic cod populations, they were also trying to correlate these fluctuations with ecological and morphological variations of these animals. Like here in the plot, in the same type of plot, you can see that in green, the populations that have a smaller population size were pointed out in other paper as the populations that also have the lower body size. But the question that remains is that true for cichlids from Lake Tanganyika that are my focus on study? In other words, there are something in the historical population size changes that can explain the current genetic diversity of cichlids, and also how the environmental and life history traits may explain that pattern. So for those of you that don't know Lake Tanganyika is located in the southwest of Africa and is the oldest and deepest of the African greater lakes. So just in a proportion, Tanganyika Lake is as long as New York. But the cichlid fishes that live in Lake Tanganyika are amazingly diverse. So far we have described 240 species that have many different types of life history and morphological traits when it comes to behavior, coloration, and so on. And that's why they're an ideal model system to answer our question. But what, in fact, do we know about fluctuations in population size in Lake Tanganyika? We know that they are potentially driven by fluctuations in the water level. So here you can see that in a variant of 25,000 years ago, we started to have a population increase, which is pointed out to be caused by the water level increase. However, this is based on only one species. The colors here represent only different populations, not to mention that we don't have any event in the ancient historical population size that could respond to the current genetic diversity of cichlids. So we lack of a broad and interspecific historical reconstruction, and we are going to do this by using relate program that uses coalescence time as inversely proportional to the population size. Just to give you an overview of my sampling design, I have a total of 620 wild-catch individuals from different collection size, brooding type, from different phylogenetic groups, habitats, and body sizes. So also a quick overview of my progress today. So basically, I sequence all these genomes, the whole genome resequencing Illumina, and then I pass all this sequencing to my bioinformatic pipeline, and then I analyze the success of this pipeline by a principal component analysis, which is exactly what I'm showing you here now. So my species, they were basically divided in three main patterns that are represented here by these species. So while the species Pseudochromus curvifrons, a literal benthic species, had clearly structured by location, the colors here represented the different locations that you can see in the map. The Bathybatis leo species, an open water pelagic species, it's actually panmictic, even though they were collected in completely opposite shores of the lake. So this is clearly driven by the ecological habitat choice. However, we also saw a third pattern, represented here by Gnatochromis biferi, where we, instead of seeing this species is also associated to the shore and deep waters. So this means that we would expect to see two clusters as we have only two locations. However, we see here a pattern of 4 clusters, which is explained by sex. So when we fragmented the whole genome into the sex linkage regions and non-sex linkage regions, we can see in the left that they completely lost the 4 clusters structuring. And this is currently are analyzing by a collaboration, but I'm not getting too deep into the details to this today. So today we're going to talk mainly about demography, and this is the plot of all my species. And then we can see, especially towards recent times, that we have contrasting trends, because in the ancient times, as they are overlapping, it's hard to see a clear pattern. But towards recent times, you can see that species such as Ben male have an exponential population size grow, while others such as Eresia have a plateau. And it's even more clear when we divide into tribes. But how can actually we interpret this? So as I said before, we already have data saying that water level fluctuations may shape. So I did a survey about the main water level fluctuations in the lake. So the first one and the most important one occurred between 60 and 135,000 years ago and divided the lake in 2 or 3 subbases. And then after that, we have a pattern of water level increased, both are followed by the African mega droughts, the last glacial minimum, and finally the monsoons activity that is currently happening in Africa. So with that pattern in mind, we can actually now interpret these patterns. So once more, I saw 3 main patterns in my samples. So while species such as Bati-Bati's leo have a more unstable population size, they respond clearly drastically to the water level fluctuations, especially here in orange, you can see the time where the lake splitted in 2, and we see that there is an immediate response of the populations with a decrease, which is different from what we see in populations such as Autoloprologus compressiceps that have a more stable population sizes and not impacted by the water level fluctuations. However, we have a third pattern of species that actually behave in a combination of factors. So towards the ancient times, they were clearly affected by water level fluctuations. However, in the most recent population size fluctuations, instead of responding with an increase of population size, they respond with a decrease, which we believe that could be due to the fact that they're actually responding to another environmental factor that we are not considering, or even due to competition with other species that had to migrate to their habitat due to their loss of the habitat. So as we see, they're clearly different, but what are the consequences of that to genetic diversity, and are they driven by ecological traits? Getting into our first question, we have to consider that we have 2 different times. So in the first time, we have the unique lake base with one determinate species living on it and an example species living on it, that in theory could have gene flow between populations, and they have the second moment, where with the water level decrease, we have 3 isolated subpopulations. And in this scenario, we clearly have 2 hypotheses: the stability diversity hypothesis and instability diversity hypothesis that I just explained to you with the B example. So when we plotted the same population size fluctuation plot, but now with heterozygosity, we expect, if the hypothesis of stability diversity was true, we expect to see a positive correlation with them. But that's not what we can clearly see. However, we do see a positive correlation with this population size stability. However, as you can see, this is very visual and quantitative. So I wanted to transform these plots into one metric that I could actually use to do correlations. So based in the Lossier paper published last year, I choose one time point, and then I calculated a delta NA, or delta population size. That is, of course, negatively correlated with the curve stability. So once I did this, I could use this steepness to calculate a linear regression, and then, of course, also do a PGLS to correct for phylogeny. So here are the results that we can see so far about the correlations. So first, when it concerns to heterozygosity. So here you can see that towards the left of the graph, we have the populations that are more stable in population size that also have a higher heterozygosity. So in other words, when we come back to our question, in the case of cichlid fishes, the stability diversity theory is more likely to reflect our organism. But coming back to our second question, is this driven by some of the ecological or morphological traits? So today I'm going to focus mainly in the habitat choice, specifically in habitat region and depth, to try to find some type of correlation. So once more, I'm here showing you a different scenario. So in the first scenario, we have 3 different populations that occupy different habitats. So for example, Batis leo lives in shallow waters, while Pseudochromis cuvrifrons live in deep waters, and we have Bentochromis microlepsis living in the intermediate part of the lake. So they're actually living, actively living in the whole lake, and they have different population sizes. However, when we have a decrease in the water level once more, some species may be more affected or differently affected than others due to the habitat that they live. So once more, we have here 2 hypotheses. The shallow waters are more unstable than the deep waters, or the opposite. So once more here you can see the correlation plots. So in the left you have the 13 carbon that will tell us where is the depth where this organism lives, and in the right the D15 nitrogen that will tell us about the trophic level of this species, and they give us different correlations. Of course, in the left we see a negative correlation, in the right a positive, but they are actually saying the same, they're stating the same thing. So pelagic species have a higher population size stability, which is actually the opposite what I was expecting at first. So when we come back to our question, our second question, both our ecological traits are pointing to the second hypothesis that states that shallow waters are more stable than deep water species. Well, I think that this can be due to the fact that cichlids are very specialized. So once they lost their habitat in the shallow water, they just migrate to the next available one. And actually the benthic species that are actually losing their habitat. And also not to mention the connectivity, because of course if the water level is decreasing like time by time and not at once, the shallow water species will be able to actually actively swim alongside the subbases. With that, I give you my take-home message. So the genetic structure of cichlids is dominated by ecological features such as habitat. 10% of my species, they were not only driven by ecological features, but also by sex-linked regions. Species with a high genetic diversity, they also present a more stable historical population size. And pelagic species, or species that live in shallow waters, have a higher stability in population size. That's it. Thank you, everyone.
Okay, kia ora. My name's Sheri Johnson. I'm an associate professor at the University of Otago in New Zealand. And today I'm going to share with you the results of the start of our research into looking at the molecular basis of thermal resilience using this New Zealand triple fin fish assemblage. So my co-authors on this study are a talented master's student, Brianna Riordan, Ludovic Dutoit, and Neil Gemmell, all from Otago, and then Tony Hickey from the University of Auckland. So triple fins are an ideal evolutionary model to study the molecular basis of thermal resilience. We have 26 endemic species that have evolved through an adaptive radiation into distinct ecotypes that occupy different habitats. So we have an estuarine specialist, a couple rock pool specialists, some subtidal specialists, exposed reef specialists, and then we have some generalists that can move between the high rock pools into the subtidal environment. So our co-author Tony Hickey has previously done a fair bit of work on some of these triple fins to demonstrate that the intertidal fish, which experience daily fluctuations in temperature, are more thermally tolerant, and they can sustain mitochondrial function better than subtidal species under acute heat stress. And so this is just showing you one of these sets of data. So it's looking at the net ATP production, and you can see here that those red and the green lines represent the intertidal species, and
They all have the same net ATP production at 15 degrees, but then as you expose the tissue to higher temperatures, you can see that the subtidal species is less efficient at maintaining that production. But we don't understand the underlying molecular basis for this thermal resilience in these intertidal species. So as a start to dig into this complex, Brianna started working on her master's thesis and asked these two questions. So using a novel fluorescent technique, which I'll describe later, we determined the mitochondrial mechanisms contributing to thermal resilience in an intertidal, the common triple fin, and an estuarine triple fin. So Fusarigian lapillum is that intertidal. It's a generalist. It can move between the subtidal and the intertidal environment. And Tony has extensively worked on this species previously. And then Fusarigian nigropenni is the sister species to lapillum. And it is an estuarine specialist, and there's been no work done on this species yet. We start with these two here mainly because they're the easiest to collect. So it didn't involve any diving or boats. We can easily just go collect them using bait catchers. And then we compare the transcriptomic profiles of the two species and look at how gene expression is altered with increasing temperature. So we collected fish from the wild, north of Otago, which is on the southeast coast of New Zealand. The estuarine fish were collected from a single location within an estuary, and the common triple fins were collected from 2 locations in the intertidal region, and we collected 80 fish of each species. We brought them into the lab, and we have a temperature-controlled system. So we provide heaters to the tank, and then we have a glycol-based thermo-regulatory system that then cools the water to the desired temperatures that we want to use. So we acclimated them to 10, 14, 18, and 22 degrees. And the reason that we chose those temperatures is because the sea surface temperature in the Otago region ranges from 7 to 16 degrees throughout the year. The glycol cooling system doesn't operate well below 10 degrees, so that was our lower limit. And then we went up to 22 degrees because there are records, of course, of those monthly mean temperatures going up to 21 degrees. We had 20 fish per temperature per species. After 4 weeks of acclimation, Brianna started to do the mitochondrial assays. She ended up doing 8 to 10 per temperature in species. These roughly took 2 to 3 hours for each assay, and so she could only do 4 fish per day, so it took her 4 weeks to complete those. And then at the end of the 8 weeks, we sampled the brain, the liver, and the heart from separate fish. And today I'm just going to show you the RNA-seq data from the brain. We did it on the heart as well, and then we banked the liver tissue. So to measure mitochondrial function, we used 2 fluorescent probes. So TMRM is a mitochondrial membrane potential probe, which of course gives you an idea about the overall health of the mitochondria. And then magnesium green, which measures the total ATP amount present in the mitochondria. So the brain tissue was removed from the fish. It was separated, incubated in these different probes. And then Tony had built these purpose-built fluorescent microscopes. And so the tissue sits on a slide. It sits on a Peltier pad. It ramps the tissue up from 12 degrees to 30 degrees. And then the computer is monitoring the change in fluorescence over time. And so for the analysis, Brianna used a segmented linear regression to estimate thermal breakpoints for the 2 different properties. And so you can see along the x-axis there you have temperature, and so we're basically looking at the points for each individual fish at where there's either membrane depolarization or the loss of membrane potential, or for ATP equilibrium when you see a decrease in ATP synthesis or an increase in ATP consumption. So here you're looking at the data for mitochondrial membrane potential. So you can see the thermal breakpoints on the y-axis. You can see the acclimation temperatures on the x-axis. You have the common intertidal triple fin on the left and the estuarine triple fin on the right. So the thermal breakpoints gradually increased with increasing acclimation temperature for the common intertidal triple fin, but there was no significant difference between the acclimation temperatures. Whereas for the estuarine species, you can see that the thermal breakpoints are significantly higher at the 18 and 22 degree acclimated fish than the 10 and the 14 degree acclimated fish. So this suggests that this estuarine specialist is better able to make thermal adjustments to the inner membrane properties or structural or physiological compensations more efficiently than the intertidal common triple fin. Here you're looking at ATP, a thermal breakpoint of ATP equilibrium on the y-axis, and again the common intertidal on the left and the estuarine on the right. And you can see for the common intertidal that the thermal breakpoints were significantly higher in the 22 degree acclimated fish compared to the other temperatures. And then for the estuarine fish, you can see that the thermal breakpoint for the 22 degree temperature is significantly higher than the 10 or the 18 degree acclimated fish, although not to the 14 degree acclimated fish. And there's quite a bit more variability in this data compared to the common intertidal. But both fish appear to make physiological adjustments which either increase the ATP synthesis rates or mitigate the proton leak from the electron transport chain as the tissue is being exposed to thermal stress. So for the differential gene expression analysis, this analysis compared the 10 and the 22 degree treatment fish only. And so on the left there, in terms of the genes that were differentially upregulated, so in the common intertidal fish, there was over 1,900. In the estuarine fish, there was over 2,600, and they were shared 690 genes that were differentially upregulated. And then for downregulated genes, over 2,700 in the intertidal fish, over 2,800 in the estuarine, and they shared over 1,000 of those genes in terms of significant downregulation. When you look at the volcano plot here, we probed the data to look at some of those genes that might be related to thermal stress or metabolic processes. So for the common intertidal triple fin, there's a couple of glycolytic enzymes that are significantly upregulated. And then some more interesting things are happening with the estuarine triple fin that are related to responses to heat stress. So there was at least two cytochrome P450 monooxygenases that were significantly upregulated, and the mitochondrially encoded cytochrome oxidase 2. Both of them had a number of heat shock proteins that were significantly upregulated. For the gene ontology, the intertidal triple fin, there was 19 gene ontology terms that were significantly enriched. These relate to defense and inflammatory response. There were some heat shock response and mitochondrial depolarization pathways that were significantly enriched, but they didn't pass the FDR. And then for the estuarine triple fin, there was 25 gene ontology terms, and a number of these were associated with metabolic processes. Interestingly, there were no shared gene ontology terms between the two species, so suggesting that they are using different physiological pathways to cope with changes in temperature. So in summary, we've shown that these two temperate species display the potential for thermal acclimation against future warming. There may be an advantage to niche specialization for that estuarine triple fin, which was better able to maintain mitochondrial membrane potential compared to the common intertidal triple fin. We use these novel fluorescent approach to study the mechanisms of thermal tolerance, and so these were built on the cheap, and they've been tested in these triple fins as well as in honeybees and looking at heat stress. And these techniques combined with the genetic analyses can help identify the processes driving acclimation. So this is just the start of us working with this triple fin complex. So ultimately what I would like to do is use this novel marine system as a test of Jensen's climatic variability hypothesis to ask whether the ecotypes that experience the least variable thermal regimes, so the subtidal specialists, the two that you see down there on the bottom, whether they possess the least adaptive potential. And so with this hypothesis, we would predict that they would have the fewest genes that are upregulated by warming and the least variation at genomic regions important for adaptation, and therefore be the most climatically sensitive to climate change. So I just want to acknowledge a number of people that assisted with this data collection or providing permission to collect samples, and I'm happy to take any questions.
Have any plans to check the proteomic landscape? Since you checked the genomic landscape and seen upregulations, but sometimes we know that doesn't translate to proteomic expression. So I'm wondering what your thoughts on that are. No, I hadn't gotten that far. For the grant proposals, we proposed looking at the underlying mechanisms for gene expression in terms of methylation or acetylation. But yeah, I'll give some thought to the proteomic expression. Yep. Yep. Yep.
Kind of on a similar line, really cool work on mitochondrial stuff. I'm wondering how that translates to higher levels of organization and ultimately fitness. Probably don't have data for it, but like how many of these fish died from different species?
None. Yeah. So, yeah, I mean, we didn't test the CT max with these, but for the northern populations, around 32 degrees is the CT max, and so you'd expect it to be lower in these populations, but these fish at 22 degrees were very happy fish, so I don't think we were anywhere close. Yep. Yep.
Yeah, so I was a bit confused about your conclusion that the common triple fin was a bit more sensitive to temperature, because it seemed like it was stable across all the different temperatures. So it would seem like the other one was more sensitive.
Oh, not that it was more sensitive, but that it was able to maintain those mitochondrial functions with the increased heat stress, more so than the common intertidal fish. Yeah. And it just may be that these two measures because Tony's done lots of work showing that that they do have more stable and efficient mitochondria. And so it just may be that these two measures in this case, we didn't capture that, or that they might be prioritizing other physiological pathways. Yep. Yep. Oh, we're done?
Yeah, sure.
Okay, yep.
I was wondering, according to Tony, the heart is probably the most sensitive tissue of the fish. So I was wondering, are you planning to repeat?
Yes. Yes, so our collaborator has done work on heart, and it's just that for us starting out in our lab where this was new and with a master's student, the brain was the easiest tissue for us to work with. But for the work that we've proposed to do, we would look at both heart and brain tissue again across all the different species. Yep.